Hello everyone, I'm Mr. Furlong, and today we're taking a look at the origins of cell compartmentalization. In the last video, we took a look at cell compartmentalization, and in this video, we are going to take a look at how that could have evolved. So let's start by comparing compartmentalization in prokaryotic and eukaryotic cells. One of the things they have in common is that they both have a cell membrane that separates the internal from the external environment. But remember, the difference between prokaryotic and eukaryotic cells is that prokaryotic cells lack the membrane-bound organelles that eukaryotic cells have. Eukaryotic cells do have these membrane-bound organelles, such as the nucleus, mitochondria, Golgi complex, and so on. So how did a eukaryotic cell get all of these things? Well, one theory is the cell symbiotic theory to explain how this could have happened. And it goes something like this. There's an infolding of the plasma membrane that then formed the nucleus, the the Golgi complex, which would explain why the cell membrane is made up of a phospholipid bilayer, as is the nuclear envelope, as is the boundaries of the endoplasmic reticulum, and so on. The prokaryotic cells, like cyanobacteria or aerobic bacteria, may have become incorporated into a prokaryotic host. Now, these bacteria then became today's mitochondria and chloroplasts. This is, again, known as the endosymbiotic theory. First off, what would be the advantage of doing something like this? Well, the prokaryotic host benefits because the cyanobacteria, which can carry out photosynthesis, could make food for the prokaryotic host. The aerobic bacteria, on the other hand, which can carry out cell respiration, would be able to provide ATPs for the prokaryotic host. So we can see the benefits of the prokaryotic host, but what about the cyanobacteria and the aerobic bacteria? What's in it for them? Well, they could have benefited because now they're protected from the outside environment, especially an environment where there might be a lot of UV rays. Hence the name endosymbiotic. All types of cells are benefiting from this arrangement. Well, what are some of the evidence that we have to support this? Well, let me tell you. Chloroplasts and mitochondria have their own set of DNA. No other organelles do. Why do they have their own set? Well, maybe it's because at one time they were their own separate organisms and they still use this DNA. If we want to make more mitochondria, that's the DNA that makes them. Chloroplasts and mitochondria are both double membrane. So when we looked at the structure of both the mitochondria and the chloroplast, there's an outer membrane and there's an inner membrane, both of which are lipid bilayers. And chloroplasts and mitochondria have their own set of ribosomes, which means that the genes on the DNA in the mitochondria or the chloroplast don't go out to the ribosomes in the cytoplasm of the cell, but they go to the ribosomes in that organelle. So at one time, all life was prokaryotic, but eventually eukaryotic cells formed. And we think this may be one way in which it happened. I'll see you in class.